The next speaker is Tanja PYHÄJÄRVI from Oulu University, and she's going to talk about genomics of clinal local adaptation in Pinus sylvestris under continuous environmental and spatial genetic setting. Welcome.
Okay, thank you for giving the opportunity to talk here. It's nice to meet so many good colleagues and the project members. Today I'm going to talk about local adaptation in Pinus sylvestris, Scots pine, and especially from the point of continuous environmental and spatial setting, where we have continuous variation in the environment and also continuous variation in the population structure. So we can start by admiring this what is considered the most beautiful, by some considered to be the most beautiful Scots pine in Finland and used in a lot of breeding. But I'm not going to talk about breeding today, but natural selection. So the theme of the meeting is this genetics to the rescue, and I just want to make this point that it's impossible to use genetics to the rescue if we don't understand the drivers and the dynamics of the past and present adaptation to the environment, and that's what I'm trying to get into in this talk. So to have adaptation, as Antoine already talked and showed quite nicely, that you need natural selection. And what we know about natural selection is that there are two big components that affect the efficacy. It's the effective population size, NE, and the selection coefficient, S. And what we know about, for example, if we start from the selection component, what is known from these Provenance trials, or as they are more widely known as common reciprocal transplant experiments, when Scots pine seed from the cold environment have been moved to grow in a more southern, warmer climates, or the vice versa, we can make conclusions about the efficacy of selection and the amount of local adaptation in this species. So using the data from so-called PYHÄ Provenance trial series from Sweden, Outi Savolainen and Timo Knur used this data to make these kind of estimates of how much latitudinal transfer. So if you take a seed from, let's say, latitude 60 and you move it to north or you move it to south, how much that affects to the total fitness. And the main pattern that you see time and time again is that in Scots pine and many other forestries, the seeds are doing best in their local environment, at least at the moment. There are deviations from this pattern in the very margins, and it's also notable that this pattern of transfer is not symmetrical. So things are a bit more drastic when you transfer to the north than when you transfer to the south. And based on the results of PYHÄ, there are some explanations what's going on here. So when you move seeds from south to north in Scots pine, the major effect on fitness comes through the mortality, and the primary cause of mortality is climate. So the seedlings get cold damage when they grow above the snow. However, when you move seed from north to south, it's what happens is that the growth is reduced and they lose in the competition. Another way of looking at the strength of natural selection in Scots pine has been these common garden experiments, where you take seeds from different latitudes, so from southern Spain to the northern Finland, and you put them in the common garden, and then you look at the known adaptive trait. In this case, it's a timing of bud set, the timing of growth cessation, where you can see very beautiful clinal variation, so that the southern seedlings stop the growth much later than the northern seedlings. And this is also very clear that there is this linear correlation between the origin of seeds and the timing of growth is also one indication of strong natural selection affecting this trait. So now we can conclude that at least for some traits, the natural selection, the selection coefficient must be high, or at least high enough to be effective. And we also know, just looking at the species distribution and amount of Scots pine trees that we see in Europe, that this must also be very high, at least in the scale of hundreds of billions of individuals. And when you put these things together, we know that natural selection in these species must be very strong. So how does this relate to genomics? So there are multiple different theoretical models for the genetics of this clinal variation. So one of Nick Barton's models predicts that you expect to see very strong allele frequency declines in a subset of adaptive loci, and then another subset of loci where you don't see such drastic differences. On the other hand, Antoine Kremer and Valérie Lecour have a series of papers showing that in some situations the selection may be actually favorable to certain allele combinations and doesn't so much change the frequency of the alleles, but the covariance pattern. So you have to look multiple loci at the same time together to see the effect. And then there are also interesting results from Sam Yeman and Mike Whitlock and from Kirkpatrick and Barton about general, like how these things are organized in the genome. And they also make some of their models predict that these genetic rearrangements and other clusters of locally adapted loci are expected to evolve. So it's easier to evolve a group of loci that are close to each other in the genome than just randomly scattered around the genome. But it's notable that these predictions actually depend quite a lot of the assumptions about the genetic architecture and the nature of clinal variation. So we don't really know what to expect, really, because we don't know the genetic architecture of this adaptation yet. So with this study, we wanted to actually look, do we see these steep allele frequency declines among Pinus sylvestris populations, because we see it at the phenotype level, or do we see another kind of signatures of geographically varying selection in the genetic data? And this was mostly work done by Jaakko Tyrmi, a PhD student. And this was started actually already in this previous EU project ProcoGen, where we sampled 120 individuals from 12 populations, trying to do like a 2 latitudinal gradients, so kind of two clines. And we used exome sequencing and obtained about 80,000 SNPs from exonic regions of the genome. So some basic background results, which were not that surprising, but this was already known from the isozyme and microsatellites and from earlier studies, but we made this repeated observation that the population structure, even when you look with 80,000 SNPs, is very weak. So if we apply this construct method that actually fits this isolation by distance model in the data, all the population structures basically explain this quite simple isolation by distance model and kind of continuous model of population structure. We also see very quick decay of linkage disequilibrium. Also, well-known phenomenon in many forestries, so that correlations between the loci in allele frequency disappear in much less than 1,000 base pair distance. So we can kind of assume that most of the loci that we are looking are actually independent from each other. So we use many approaches to look at adaptive variation, but I'm presenting here two main approaches. So we wanted to identify these parallel allele frequency clines in the east and western cline by very simple regression analysis. Luckily, because the population structure is very weak, we don't have to actually use very complicated models in this case. And then we use these similar models that the previous speaker was using, like the space scan and such, that actually are based on identifying exceptional divergence among populations, but do not explicitly have this clinal variation built into the model. So the regression analysis looks like this. So we have a latitude of the population on the y-axis, and then we have allele frequency, on the x-axis, and allele frequency in the y-axis. And then we can either look the whole data together, or we can look the eastern and the western parts of the allele frequency cline separately. And we use this generalized linear mixed effect model and just ask a simple question: does a model with the latitude explain the allele frequencies better than the model without the latitude? And to cut long story short, we don't find any outliers at the false discovery rate of 10%. So most of the loci look like this. And our most extreme locus looks like this, where we go from the 0 to 0.5 frequency, but considering that we are looking at 80,000 loci, this could be just a result of chance. And then this base scan, which is identifying the loci that are extremely diverged across populations, and where selection is inferred if a locus-specific component is required to explain the observed allele frequency pattern. So basically, that one locus deviates strongly from the majority of the genome. At the Q value, cutoff 0.1, so again 10% false discovery rate, we actually find a single outlier among the 80,000 loci, and it's luckily in non-coding area, so we don't have to provide very lengthy explanations what it could do. And it's worth noticing now that if you look at the actual pattern, it's probably driven by these three populations here, where the allele frequency is much higher than in the other populations. And this method is not actually designed for identifying clinal patterns. So we see differentiation, but it's not actually anywhere near what we would expect from the clinal variation. But when we started to look into this SNP in more detail, we found out that there were actually other 43 SNPs that were in very high correlation and had a very high linkage disequilibrium with that outlier SNPs. And they were distributed in 25 different scaffolds. And this kind of caught our attention because we know that this very strong linkage disequilibrium is very rare in this genome. And all these SNPs actually have very high or relatively high derived allele frequency in one Eastern Ust-Kulon Russian population. And when we look at this, we kind of infer the haplotype. And when we look at this haplotype, we notice that within this haplotype there is very low diversity, so about 10% of the other haplotype. And it has a very much higher differentiation between populations than the genomic average. And we thought about there are many potential explanations for phenomenon, but we think that most likely explanation is that there could be actually an inversed part of the genome in linkage group 1 in this Westbrook et al. Pinus theta map, because almost all these 43 SNPs that we can put on the map go to the same linkage group. And we think it might be a very big chunk of the genome that is going maybe through a selective sweep in a subset of populations. So to kind of demonstrate this effect, that this is the measure of linkage disequilibrium, R square, and it's about 0.5 in the SNPs in this putative inversion. And if we make permutations across random scaffolds with the same amount of SNPs, the linkage disequilibrium is much lower. So this group definitely is different to the genomic average. So in summary, Pinus sylvestris has strong phenotypic clinal patterns, and a large population size and strong selection should make natural selection efficient. But allele frequency declines are very rare in Pinus sylvestris coding regions. But the joint analysis of these multiple SNPs reveals this unexpected linkage disequilibrium between loci. So we think that there might be a very big inversion in this genome. And in the future, we want to develop better methods for looking at the multiple loci at the same time, and also to start exploring non-coding genomic areas, and also looking at this mapping of this inversion haplotype better to actually look whether it's inverted or not. And at the end, I would like to thank all the funders, collaborators, and co-authors, and especially Jaakko Tyrmi and Outi Savolainen, who contributed a lot to this work. So most of the work is actually done by Jaakko, so I'm just presenting his work.
So thank you, Tanja. So we have 5 minutes for questions.
Hi, Tanja. This is Andrew Eckert from Virginia Commonwealth University in the United States. I have a question about your linkage disequilibrium analyses. You matched the permutations on numbers of SNPs, you said?
Yep.
Correct? Did you also match by allele frequencies of those SNPs for R squared, which might be important?
I would have to check.
Okay.
I know what you're after, because if the allele frequency would be very different, I should check. I think we thought about it, and Jaakko probably did it, but I would have to check.
All right, thank you.
Yeah, Santiago González from New. I was wondering if, can you tell more about this inversion? Is present in other conifers, or there is something you can get from comparative mapping there, or it's just very early times for that?
So there is a comparative map between sylvestris and theta, and it's not obvious there, and from the cytological cytogenetic studies show we know that the inversions and rearrangements in conifers are not very common. So that's a big kind of why we want to check whether this is real or not. But the other explanations are even more weird, like that we would have a population-specific region of low recombination, or 350 megabase per area increased by sweep. So, and of course there is always possibility for a technical mistake, but Jaakko has gone and checked all the alignments by just visually and doing a really careful check of the data. So it will be really interesting to actually check whether it's an inversion or something else, or did we just make a mistake.
Hi, Tanja. Juan Pablo Jaramillo from UNAM in Mexico. With this inversion story, you just made me think about our friends working with teosynths, who have discovered some inversions who are adaptive to altitude in teosynths. Now, my question is if you have had the opportunity to look at trees growing on field test, how they are behaving. If you compare, the fitness or the budget times with the genotypes of these potentially adaptive SNPs of yours?
Unfortunately not, because this, where we see these haplotypes are the eastern populations, and they are not very often included in any of our common garden studies or anything. So we really should put more effort on including those eastern samples into the analysis. But yeah, that would be, cool experiment. I know that in Teosint they are doing these experiments now and showing some cool results.
Okay, so I think we have to move on. Thank you, Tanja.